Hello, my A&P1 lab students. How are you? I hope you're doing well. So, we're here today to talk about, actually, let's call it, it's lab 3, exercise 5, cell transport mechanisms and permeability. So, we're going to talk about passive processes or passive transport. Just get my pen here. Can't live without my pen. We're going to talk about in the passive type what is diffusion and the different types of diffusion, simple, osmosis, and facilitated diffusion. And then we're going to talk about filtration super quickly and about active processes, active transport. Okay, so I want to start by telling you what diffusion is. Diffusion is molecules moving from where they're more concentrated to where they're less concentrated. Which molecules? Any molecules. Depending on the molecule, you're going to have a different type of diffusion. Okay? So if I show you this, look what a nice image. What do I see here? I see small particles and big particles, right? So let's say we're talking about the big red particles. Which side do you see more particles? Left or right? There are more particles on the right side, right? Okay. Now look at this membrane here. You see this here? That is your membrane, right? That is your membrane that is selectively permeable. So we're talking here already about selective permeability. What membrane in the body is selectively permeable? If you said plasma membrane, got it. Plasma membrane, right? So the plasma membrane of your cells have ability to allow some substances to pass and not allow other substances to pass. One of the things that stop some particles from passing is size. So that's why I'm here with the big balls here, with the big molecules. So they're so big that if one was here, they would not be able to pass because of their size, right? Okay, so what happens? How can we equilibrate these two sizes? So first, what's equilibrium here? We're talking about concentration, right? Concentration is what? Mass per volume, right? What is the mass here? The red molecules, whatever they are. What's the volume here? Water, right? So, if I cannot pass my red molecules to the other side to equilibrate how many red molecules are on one side and comparing to the other, if they cannot pass, what can I allow to pass here? I could allow water to pass because water is really small. So then water moves this way. I'm not calling these blue molecules water. I want to do something else with them, okay? So let's say all this beige area here is filled with water. So water is going to pass this way to do what? To try to dilute these red molecules. So then remember that concentration is mass per volume. If I increase the volume, is the same thing as if I was decreasing the mass, right? If you divide one by the other, they are inversely proportional. Does it make sense? Inversely proportional. So, if I increase volume, am I then equilibrating the concentration, not the number of particles, but the concentration, which is mass per ml, or I don't know, grams per ml, that's concentration, right? Would I be doing that because I'm adding more water and I'm diluting this, and that would be equivalent to having less red particles in less water? So, in summary, a lot of particles in a lot of water would be the same in concentration of a little bit of particles in a little bit of water, right? think about a cup with dye, and then you have two cups, right? One super big and one small. And they're both the same tone of, let's say the dye is pink. Which one of these cups is gonna have a bigger concentration? The big or the small? If the color is the same and the color of dye is gonna give you an idea about concentration, they're gonna have the same concentration, right? A lot of dye in a lot of water, or a little bit of dye in a little bit of water, in the end, mass per volume would be the same. That's the point here. What did I just teach you? Yes? Did I just tell you what osmosis is? Yes. It's the transport of water to where solutes are more concentrated. Period. And what is more concentrated? You're always comparing to another region, right? So why am I showing something here that is just a square with a division in the middle, and I'm calling that selective permeability or selective membrane? Because this is what's going to happen with the cell compared to the outside of the cell. And you may think, well, but which one of these is the cell, the right or the left? It could be either one. It depends on the particle. It depends on where there is more concentration, it would depend. Okay? But making this in these two squares, it just makes it more clear for you to understand how this plasma membrane is formable. Okay, so if now you know what is diffusion, I just want to erase all this. I want to show you something else. Now that you know what is osmosis, which is a type of diffusion, now I'm going to tell you something else. So now imagine that I have these little blue molecules here, right? And they are just a different type of molecule, a different solute than the red one. Do you agree that their size is small enough that they can pass through the selective permeable membrane? Okay, so if they can pass, which direction would they go? From left to right or from right to left? Try to think about that. What is diffusion? Is go from where they're more concentrated to where they're less concentrated. How do you know they're more concentrated here? Because the volume is the same in both compartments and there's more particles here. So they go from here to here. That's the direction. Does it make sense? I hope it does. So this is what we call simple diffusion. So what you could ask, so why don't you talk about osmosis here? You're still gonna have water moving both sides because water is small enough to pass through the channels, right? There are little channels in the cell for water, and they can even pass through the plasma membrane. So why is it not osmosis? Because osmosis mostly happens when the particle that you were talking about, which was my first case, is non-diffusible. Non-diffusible. I think diffusion is one F. I think I'm misspelling it. Diffusible. So when the particle is non-diffusible, that means you need water to move in order to change the concentration here, right? Because the particle cannot move through that membrane. If the particle can move, then you're not going to have so much of an osmosis effect and more of a simple diffusion effect. Got it? So that's the difference between the red and the blue particle. Okay. Now let's talk about another situation. For some reason, and there are many reasons why in the cell you need this, and the main one is sometimes you need the polarization in the membrane to happen, and for this membrane to be polarized, you need a difference in charge comparing the outside and the inside. And in cases like that, you may need, not the red one, but let's talk about the blue one. We may need the blue particles to go this way. And what is that way? That is against the concentration gradient. So if you're going against the concentration gradient, do you agree that it's like you trying to paddle a boat in a river against the stream? Are you going to need a lot of energy for that? You're going to need a lot of energy. So what do we use here as a source of energy? Please tell me that you were absolutely thinking about ATP. So the channels that allow molecules to move against the gradient to make the left side even more concentrated than it was already. That is the type of channel or carrier that requires energy to do that transportation. Okay? One example of this is the sodium-potassium pump. It's an ATPase pump. Means what? It needs ATP to be broken so sodium can move out and potassium can move in. Okay? Because there's always more sodium out of the cell. So we're going to learn this in the lecture, right? I'm just saying here, and you're going to listen this in a lot more details in your lecture. But because you are moving sodium out of the cell where there's already a lot of sodium, that is called moving against the concentration gradient, which is when you use to break down ATP to generate energy. Okay? Okay, so the process where you go against the gradient and you need ATP is called active. Process or active transport. Okay? So this is not diffusion, it's active transport. You're going against the concentration gradient. So I'm going to go back to my first slide where I say, let's see if we agree with these definitions then. We said what diffusion is, and now what is simple diffusion? Solutes dissolved in water that go along the gradient, down the concentration gradient, from more concentrated to less concentrated. Osmosis, the transport of water, and I would add here, when the solute is non-diffusible through the plasma membrane. What is facilitated diffusion? It's just a diffusion that doesn't happen through the plasma membrane, because usually molecules that are liposoluble just diffuse through the plasma membrane, which is lipidic. But if you're transporting glucose, proteins, ions that have charges, or molecules that are a little bigger like a protein, then you need a channel or a carrier protein. Okay? What is a channel? This is a channel. Look how pretty. This is a channel embedded. So this is the plasma membrane. This green here is the channel. And the molecule would go through here. Okay? That's a channel. And a carrier is just a similar type of molecule, but there is like a little place where the solute binds to be carried out or in. It's similar, okay? Well, filtration is another type of passive process. It's just when water and solutes are forced through a membrane by hydrostatic pressure. This is something that you see, for example, when we study the kidney. You're going to understand how the glomerulus has that type of effect, okay? You also have this type of effect, for example, in plexuses that are part of the structure of your eye, and when you filter blood and you press the blood against the endothelium of those little capillaries, just part of the liquid is filtered out of the capillaries into a space, a segment of the eye, and that's going to be just a transparent liquid. So you're like pressing that blood against the blood vessel, and something comes out on the other side, which is out of the blood vessel. If you didn't really get this, don't worry. I'm just telling you, filtration is something that also happens in our body, okay? And you're going to learn this in the future. I'm not going to require you to know these examples right now. What else I want to tell you here? Passive active process transports. I'm going to post 2 videos for you about vesicular transport. So we already talked about the one that requires energy as ATP, but I'm going to post a video about the other 2 types, which are endo and exocytosis. Okay. All right. So moving on. Now I'm showing you diffusion through the plasma membrane. So now you see that this is the phospholipid bilayer from the plasma membrane that we learned last week. And this is the outside of the cell, this is the inside of the cell. And so this is just a piece of the cell, right, that you're seeing. And what is going in? These green molecules are going from where they're more concentrated to where they're less concentrated. Well, what if they do the opposite? If they go against the concentration gradient, they need energy. What is the form of energy we use? ATP. And here is just to show you what I just said about facilitated diffusion. Either you use a channel, and this is a channel here, or you use a carrier, and this is a carrier, okay? They're very similar. When your molecules are small enough or lipidic enough, they can pass through the plasma membrane. You may ask, how does water pass? How does water diffuse in and out of the cell? Either through a channel that is called aquaporin or through the plasma membrane, okay? All right, now I have a video for you about diffusion through the agar gel, so you don't need to worry about this in this video. You just watch the next video. It's just like a 2-3 minute video for you to understand what is this experiment that we would be doing in class. And this is the diffusion through water. It's another experiment with doing lab, where we're going to add 1 drop or 2 maybe of a dye, of a concentrated dye, in water. What happens through time? So time here. Time is passing. And as time is passing, that dye that is super concentrated here and not here tends to go from where it's more concentrated to where it's less concentrated, which is everywhere else in the water. So what is the tendency and what's going to happen in the end? So this is the end of the experiment. The dye is going to be dissolved everywhere. You're like, okay, we saw this before. This is just to remind you how diffusion happens, okay? All right. Now we already talked about diffusion through living membranes, and there is an experiment that we do in the lab. So the experiment is, we are going to, first, our cells have 0.9% of sodium chloride. Okay, that's the concentration of sodium chloride inside our cells. If you add this cell to, and then the experiment is done with red blood cells, okay? We add red blood cells to an isotonic solution. What is an isotonic solution? Solution that has the same concentration of whatever ion or whatever substance or solute than the inside of the cell. So if you add, so if it's 0.9 here, and you're adding a solution that, so the solution is in the container, right, in the square here, that is also 0.9%, what's going to happen to water? And why are we looking at water here? Because water is what happens first. So if you put the cell in like a shocking condition, first water is going to move. And then you may have movement of solute, but here, because it's kind of shocking, the next situation is kind of shocking, the cell doesn't have enough time to diffuse the ion. So you see water moving first. What happens here? Water is moving freely both ways. The cell is going to be happy forever. Nothing happens. Okay? Just movement of water in both ways in the same amount. Isotonic. Now imagine that you put this cell, this red blood cell, in a solution. So the cell, again, the cell is 0.9 and you're putting it in a solution that is 10% of sodium chloride. Okay? What's going to happen? Hold on a second. All right. There was something that I didn't really like, so I just wanted to erase so you don't get confused. Okay, so I was telling you, not hypertonic, hypotonic. That's a different situation. You have 0.9% of sodium chloride inside the cell. What is outside of the cell? Water. It's like 0% of sodium chloride or 0.000 something, right? Super low concentration of sodium chloride. What's going to happen to water? So that blue arrow is the water movement, just like here, right? So water is going to want to go to where it's more concentrated in solute, so we can dilute it, right? So water goes in, and there's a little bit of water that goes out, but most of the water is going in the cell. What happens? Lysis or hemolysis. When you talk about red blood cells, you can talk about hemolysis, right? Cool. Now we have another situation where it is hypertonic. What is hypertonic? The solution that you're adding has a lot of tonicity, it has a lot of sodium chloride. Here is the 10%. So again, cell is 0.9% of sodium chloride, and outside of the cell is 10%. It's a lot different. So in order to equilibrate the amount, the concentration out of this cell, what water from the cell does? Water from inside your cell is going to move out in a hypertonic solution. That's what you need to know. And the term for this is crenate or crenation, okay? So here, what's going to happen to the cell is the cell is going to lysis by bursting. And here, the cell is going to shrink because it loses water and it looks like a little raisin, right? Okay. In this situation, usually you can see the cells. Let me see, I think I have pictures for you. So this is what happens in the isotonic solution. The cells, these are red blood cells. They're healthy. This is how they look under the microscope in a real world type of scenario. This is what happens with the swollen cells in the hypotonic solution. So we say that we find what we call ghost cells. And if you want to know how long this takes to happen, it's like seconds. And this is the hypertonic solution. See how the cell shrinks, and look how they look. They look like a little raisin. Okay? So we talked about diffusion. We talked about tonicity and cell permeability. We talked about the red blood cell experiment. And now I just remind you to watch the video about the agar diffusion and the one about endo and exocytosis. Before I go and leave you alone, I just wanted to show you one thing. Remember how this week we have a lab report, right? So what is the lab report that you're going to do? Let me make this smaller so you can see the real thing. So I am in the Mastering A&P website. Okay, so you're gonna click on your Mastering A&P website and open and put your password and go in. Click on our lab course, and then you're going to have to go on this left column to study area. So when you click on study area, you see this page, and then you click launch the study area, and then you're gonna see this page. Nope, you're going to see, let me go back, let's launch it again so you see exactly how it looks like. So when you launch the study area, you're going to see this page, And in this list, you can use all this to study, okay? But you're gonna go to PhysioX. And now you're gonna see this page. So now you're gonna click on exercise. And now, exercise 1. Is that what we need to do for week 4, the lab report? Right. So which of these 2 experiments do we need to do? 1 and 3, right? They're very similar, but one is going to be focusing on simple diffusion and the other one is going to be focusing on osmotic pressure. So you click on it and then you can't really see here. Let me show you the whole screen. You're going to read and move on and read and move on and answer. So if you don't answer these questions in your lab report in the end, it's going to show empty that you did not answer. So you have to answer. Because you could skip like I'm doing, but you need to answer, right? And then you just go submit and then continue. And this is the experiment, right? And as you do, you have to do, how do you do this experiment? I have no idea. You only have to follow directions. And the directions are in this blue box. Read what it says and do what it says. Drag the 20 MWCO, the membrane, 20 molecular weight, to the membrane holder between the 2 beakers. So these are the 2 beakers. Which is the membrane 20? I'll just grab it. Oops. Here. And then I click. You can't see here, but you click next. What is telling you to do next? Increase the sodium chloride concentration to be dispensed to the left beaker to 9 millimolar, selecting the plus button. Sodium chloride, 9 millimolar, dispense. And that's what you're going to do. So in the end, what's going to happen? So you see you have the experiment, you did the pre-lab quiz, you do the post-lab quiz, review sheet, and lab report. When you get to the end, it's going to ask you, do you want to print and save your version? And then you put your name here, and then you click OK, or you can say lab report, whatever. Print and save as, and it's going to show you here everything, the information about the report. I didn't do anything, right? So it's very short report, see? But you're going to do, and this is what's going to happen. And you're going to save this as a PDF. What are you going to do with this PDF? You're going to upload in week 4 in a Dropbox in iCollege. Is it clear? If it's not, go back and watch the video because I don't have anything else to tell you about this other than follow these directions, okay? So this is how you're going to do this lab report, and then we have 2 other, for today's exercise, you're going to do this for exercise 1, and then you're going to generate another report for exercise 3. And then in following weeks, we have 2 more reports that you're going to click on PhysioX, look for the exercise, all the same, and do a similar thing. But I'll send you more videos about it, okay? But you know what to do now, okay? Bye. Good luck.